So we have a regulatory perspectives panel coming up as the panelists are making their way off the stage. I'm going to start by inviting our colleague from Japan, Hide Nakamura, to the stage. He's going to frame things with a short presentation, and then I'll invite the rest of the panelists to join us on stage. So Hide, take it away.
Hi, I'm Hide Nakamura from National Center for Child Health and Development in Japan. 2 years in a row, I'm in a session with my colleagues from FDA. I'm not from FDA or PMDA, but I have experience as a reviewer before. So let me give you some examples of what I experienced. And 3 of my slides talks about if the draft guidelines or publication. So if I say something wrong, please make correction later. So next slide, please. So, when we look at rare, each physician see very small number of patients for rare diseases. So it's like, blind monk touching different parts of elephant. So if the draft guideline says identifying patient population, identify or develop clinical outcome assessment, identify or develop biomarkers, design externally controlled study. These are the important things. And natural history study also benefit patients. Next slide, please. And this is from the draft guideline. These are different aspects which is important for developing drugs for rare diseases. Again, natural history, disease pathophysiology, clinical manifestations, and identification and use of biomarkers. Now, biomarkers is probably one of the keywords we'll be discussing about. Non-clinical studies, efficacy endpoints. Again, I'll show you some examples of how difficult it was. Evidence of effectiveness and safety, pharmaceutical quality consideration, and some additional considerations. And FDA guidance is almost always our interaction with FDA. So next slide, please. So now I'll show you some examples of what we experienced. We did clinical trials on MELAS, mitochondrial encephalopathy with lactic acidosis and stroke. And the clinical trials for 2 years couldn't show the efficacy. And primary endpoint was a rate of improvement of stroke-like symptoms. And so have to decrease the number of strokes during that period. And well, we couldn't show the difference. Well, I should say the specialists who did clinical trials couldn't show the difference. Maybe small number of patients, maybe clinical trial period are too short, or inadequate endpoint. And I'll show the next slide. When they started these clinical trials, they didn't know there's a juvenile type and adult type whose symptoms and progress are different. Next slide, please. No, I didn't. Yeah, and that. But after, so they continue to do non-GCP observational trial for a long time. And usually these kind of patients will become bedridden, but no patients went to bedridden. And they, the specialists, brought it back to PMDA, but PMDA wanted more solid information for like a natural history. And for this kind of rare diseases, doctors do survey, like a national survey for every 5 years or so. And they couldn't look at all these every year information. So PMDA, they never could submit enough data for PMDA review. So next slide, please. And left side is this Allegheny study. This is a definition of stroke-like episodes. And that's very important for this primary endpoint. On left side for arginine study, and right side is for taurine study. And taurine was actually approved by PMDA. Taurine study was started later than arginine study. Most of the members involved in taurine study was also involved in arginine study and knew that there's an adult type and juvenile type and adult type. So maybe the difference of this, if you look at this carefully, it's very different. Scores can be like the number of stroke-like episodes could be very different, and so difference of endpoint may cause the difference. I should say may be the reason for the failure of arginine study. Next slide, please. Oops. Here. Oh, here. So this is a survey, and see these two peaks, and symptoms and the progress of the symptoms are different between these two types. Juvenile type often progress very quickly, and patients become bedridden. Adult type progress more slowly. Next slide, please. And for this MELAS, they used scores, and they have like 20 different things to score and add them up, and they want to look at the severity. But if you look at scores of rare diseases, it's not like really linear. And if it's linear, it's easier to evaluate drug efficacy. But if it's not, so looking at where the drug works, you may not be able to use a score for drug evaluation, and that could have been one of the reasons why we couldn't also show efficacy in secondary endpoint. Next slide, please. This is from another clinical trial. When we did enzyme replacement clinical trial on enzyme replacement therapy for type 2 micropolysaccharidosis, before we started, we asked the doctor to show this is the age and the psychological development using what they call Kyoto scores. And if the child develops properly psychologically, it goes up linear. And turned out that the psychological development start to deteriorate around 2 to 3 years old. So if we do drug study very early, it's hard to show drug efficacy. So we decided, usually we get grants only for 3 to 5 years. So we decided to pick up older age groups to show the efficacy of this enzyme replacement therapy. Next slide, please. And another example is familial intrahepatic cholestasis. And liver function deteriorates fairly quickly, and most patients get liver transplant. Next slide, please. And we did this study. It's buffenil, and doctors said most patients get liver transplant within a couple of years. And I didn't believe it. So I asked them to do the national survey. And next slide, please. There's a large variability of disease progress. Some patients get liver transplant within 1 year. Some patients get liver transplant in 10 years. So, well, if patients get very sick quickly, true endpoint is avoid liver transplant. But it's difficult, and serum liver function tests could fluctuate. Itching may be not good enough. So we ended up establishing and validate new endpoint, and it's a pathological endpoint, validated, and it's published. And well, and these three we discussed with PMDA. This clinical trial is complete, and now it's ready to submission. There's some political issues, so it's a delay, but it's ready for submission. So next slide, please. And last slide, this is from FDA's review. we hear biomarkers all the time. That we got to be careful. Like academicians tend to say, "Oh, this is biomarker. See, this is a statistical correlation." It's not the case. We have to define what kind of biomarker it is. Is it diagnostic, prognostic, predictive, or responsive? So when we discuss about biomarkers, we have to specify which biomarker we are discussing about. And this is the last slide. And I think hopefully this can give you a better impression of what we are discussing about. Thank you. Sorry, it's longer than 10 minutes.
I want to thank Dr. Nakamura for his opening comments. I think he's really set the stage very well for the conversations to come here. So if we could get our panelists to come up, Dr. Jeff Siegel, Dr. Celia Witten, Dr. Lauren Wood Heiickman, Dr. Anita Zaidi, and of course we have Dr. Nakamura already with us.
Excellent.
So I think everybody can see the complexity of developing drugs across the wide range of ages from pediatrics through adults. I think we've often heard that kids aren't adults, but the converse is also true. Adults aren't kids. And so I think we have to think about it fairly broadly when we want to think about drug development for children and where we start. So I'll turn it over to Klaus to get the conversation.
Sure. And Hide, you set it up perfectly because I'm going to build up on where you concluded. Understanding exactly what is the intended application of a potential biomarker in drug development. I may start with Jeff because you see that from a unique vantage point across multiple indications. So if you could comment on that perspective of really understanding the intended application of those biomarkers.
Sure. Thank you, Klaus. So my name is Jeffrey Siegel. I'm Office Director for the Office of Drug Evaluation Sciences at the FDA in the Center for Drugs. The qualification program for biomarkers and clinical outcome assessments sit in my office. So when we think about validating, qualifying a biomarker, we think about two things to help guide the development of evidence to support use of the biomarker. We think about the type of biomarker that it is and the context of use. So the different types of biomarkers, you heard from Dr. Nakamura, they can be prognostic biomarkers, they can be biodynamic biomarkers, in some cases they can be surrogate endpoint biomarkers. And in the context of use has to do with the way that the biomarker is going to be used in a clinical trial, in a clinical drug development program. So, for example, a prognostic biomarker might be used as an enrollment criteria to enrich clinical trials in patients more likely to develop an endpoint, to allow studies to be shorter, small, and smaller. A biodynamic biomarker might be used for choosing a dose for phase 3 trials. And a surrogate endpoint biomarker might be used as a primary endpoint for approval of a new drug. So it's very important to consider the type of biomarker and the way it'll be used in clinical trials. Then with that information, we think about the analytic validation and the clinical validation. So, and these depend very much on the context of use. So the analytic validation has to do with the sensitivity of the biomarker, its specificity, the reliability and accuracy. And then the clinical validation has to do with showing that the biomarker reflects the clinical concept of interest. So for a prognostic biomarker, you need to determine how well the biomarker selects patients who are more likely to have more rapid progression, for example. For a surrogate endpoint biomarker, considerably more evidence is needed because this is going to be used as an endpoint for approval. And one last consideration, we think about the benefit and risk of use of a biomarker. And when we talk about benefit and risk, we're talking about something quite different than benefit and risk for a drug. For a biomarker, the benefit of using a biomarker is its benefits to facilitate drug development. So does it help do trials more quickly with smaller numbers of patients? The risk has to do with what the consequences are for patients if the biomarker actually doesn't measure what it purports to measure. So think about a surrogate endpoint biomarker that's going to be used for drug approval. If the biomarker actually doesn't predict the clinical outcome of interest, then you could end up with a drug that doesn't work and patients would be exposed without experiencing any benefit.
Great. I think our next question is for Dr. Zaidi, and I'm probably mispronouncing your last name, so please feel free to correct me. But the agency encourages people to come early in discussions and to have repeated conversations. And I think that applies not only to medical products, but also to some of the tools that we use for development. Could you comment on some of the mechanisms that the agency has for engagement around biomarkers, clinical outcomes assessments, and other tools? Thank you.
Yeah, thank you. It's Anita Zaidi, but that's okay. And I just want to apologize. I have 2 kids in daycare, so I'm sick every other week. Yeah, so we definitely encourage early involvement with us early in the clinical development space. Please, as soon as you have any ideas or an inkling of a clinical development plan, please come to us. We have many tools. And, that's what we're here for, is to provide advice and to help guide you through the regulatory process, because it can be very complicated. And I think, what I've seen in the lysosomal storage disorders, which is, what my division focuses on, is that we have a lot of clinicians and a lot of investigators that are, and we're really, we applaud anybody who's looking at, lysosomal storage disorders for development, because it is such a difficult space. And so what we've found is that, coming to us early, we can help kind of guide you through this whole process, not only just with the clinical development, but also the non-clinical development and the chemistry and manufacturing, things like that. In regards to the clinical development, specifically for lysosomal storage disorders, a lot of focus now is on the neurologic endpoints. And so COAs are really important in that regards, because we need to know that, we want to look at things that are clinically meaningful to patients. So we actually have the COA division, and they have actually made a lot of strides in the past few years about really providing very meaningful advice when looking at COAs. One is to make sure that the COAs are meaningful to the patient. That's obviously the most important thing. 2 is to make sure that they're looking at COAs that are valid and that they're reliable, and that they can provide the data to support that. There's a lot of COAs that are looked at in the clinical space. And so, the question is, how do we move, the COAs from the clinical space into the regulatory space, into the clinical trial design space? And so we have the COA division that can help really move that forward. And then in regards to kind of the biomarkers and looking at surrogate endpoint, we actually, I think Dr. Siegel is part of, there's a surrogate endpoint meeting, the type C, that you can always request when looking at that. But again, all these things need to really be done very early in development, because we can help, because we're going to ask for data. We're always going to ask for data. And so that needs to be done early so that you can start providing the data, and then we can start designing, pivotal trials that are going to be clinically meaningful and can show, safe and effective drugs. The last thing we want is a trial to fail not because the drug doesn't work, but because the design was wrong. So, thank you.
That was a great perspective from the CEDAR angle, but I'll go to Dr. Witten next, because you've been pretty visionary in the way you have helped us structure the collaboration around alpha-1 antitrypsin deficiency. And I wonder if you can comment on from the CBER angle on all these aspects of truly understanding and defining how specific biomarkers can be used for specific applications in the drug development continuum, and how we're partnering with as many groups in the agency as possible to make that happen.
That's kind of a tall order for an answer to one question. So maybe I'll just pick a few topics and comment on them. And first, I want to say, I think Dr. Nakamura's talk was great in describing some of the key issues that we face, including with alpha-1 antitrypsin. He gave an example, alpha-1 antitrypsin deficiency for pulmonary disease. He gave an example where the pediatric version of the disease was worse, and that there was a nonlinear relationship between the biomarkers and the clinical course because of that. And I think the, pulmonary function for alpha-1 antitrypsin deficiency and a lot of other diseases too, where there's a lot of reserve, someone can get worse in the early years for quite a while before that really shows up clinically. And which is the same problem Dr. Nakamura was mentioning, just at the other, just flipped around. And I think for not uniquely for alpha-1 antitrypsin disease, pulmonary manifestations, but a lot of other kidney disease and retinal disease, a lot of other things that you could mention. Sometimes we've had trouble finding really effective treatments later when the clinical manifestations are already there. But it's challenging to figure out what to do earlier in the, how to study it earlier in the disease. For one thing, we need to understand better who's really at risk to progress, because these diseases are quite heterogeneous. So an understanding from biomarkers about who would progress would be helpful in figuring out who to treat early. That's one question. And then how to measure the benefit of that is also a challenge. And our way of looking at these a lot of times is in a way that is assuming linearity, that we're looking for a linear relationship between the biomarker and the clinical manifestations. And if that, we know that isn't met, so that is not the way that we need to go about this. So I'm not sure what it is, but I'm just saying that is an agreement. I'm just agreeing with what you said, Dr. Nakamura. I think that's a challenge. One challenge across the lifespan, because you might want to treat early, but we do treat late, and maybe that's not even possibly going to have any chance of success. I'm not saying doesn't, depends on each situation. I want to mention maybe it's not exactly a question across the lifespan, but it's just one issue about biomarkers that's important in the gene therapy area that may not sort of come up. So it's not really the answer to your question, but I just want to get a chance to say this. But one thing is we do look at a lot of times biomarkers, and we're looking to see whether that's, we want to look at that biomarker to include or exclude a patient from a trial or from a treatment. And that gets to what Jeff was saying about clinical validation of those kinds of biomarkers. And there's a challenge in doing that. So for gene therapies, sometimes we will want to look at immune response to the vector or the transgene. And it's not clear, so we'll have some way of measuring that. And then it's not clear whether we should exclude those people from the trial or whether we should include everyone in the trial and then figure out a target level. So that's a challenge that we commonly face, which involves the use of biomarkers. And I think it's particularly acute in the gene therapy area.
And that's a fantastic point related to the question, because at the end of the day, as Jeff was pointing out in his talk, it's really important to understand the ramifications of the application of a given biomarker for a drug development context. And so, Joe, do you want to go to the next question, which I think is a really relevant one to continue the conversation?
Yeah, I think so. I think Dr. Nakamura, following on your talk, one of the things that you alluded to was the idea of how important optimal patient selection is. I wonder if you could elaborate a little more in thinking about the screening strategies that might need to be employed in order to optimize your patient selection and find those patients.
Well, it's a difficult question.
Yeah.
Well, in that particular case, well, if we really want to save that particular case with psychological development, if we want to look at drug efficacy in a short period, we probably so for that purpose, we started the trials for older ages. But if we think about care of children, the proper development of children, we have to start from every birth. So to do that, we need a good biomarkers. So I think finding better biomarkers for the discussion about gene therapy and regenerative medicine in neonatology, we really have to find a good biomarker to predict the outcome. And we don't have that many. And that's why we really have to look at the natural course, the natural history of rare diseases to make sure what each physician have different impression because there's such heterogeneity. So we really need to have a very good number of patients' information to see, how disease progress.
Great, thanks. That's a good point. I think that really leads in then to a question for Dr. Wood Heiickman. The type 1 diabetes community has quite a robust understanding of the natural history of the disease. I wonder if you can elaborate a little bit more on how important these staging paradigms are in terms of supporting drug development, and what is the evidence that the agency would like to see to support those staging paradigms.
Great. Hello, everyone. Thank you for having me today. My name is Lauren. I'm a medical officer in the Diabetes, Lipid Disorders, and Obesity Division. And I think that we're extremely lucky in type 1 diabetes with the well-developed research paradigm for supported by longitudinal studies from early life cohorts over decades that support this staging paradigm. And from my perspective, I'm a medical officer who was involved in the review of the first disease-modifying therapy for type 1 diabetes, Tzield, which is approved in children 8 and up, children and adults 8 and up at risk of developing type 1 diabetes. And the staging that was the groundwork that was laid by academics and the sponsors of the drug program really set the stage for our regulatory review and led to some really important considerations for benefit risk. A little background on type 1 diabetes staging. They've actually founded this research over decades, and there's one great thing is that it has a consensus statement. There's in 2015, the JDRF and Endocrine Society support the staging paradigm, and then also the robust research framework. In addition, the autoantibody, so there's a couple of biomarkers that go into the staging paradigm. And whenever patients are diagnosed with stage 1 type 1 diabetes, they have 2 or more islet autoantibodies to their pancreas, and that harkens the beginning of autoimmunity. And then whenever they start to show signs of organ dysfunction, they're diagnosed with stage 2 by glucose testing on an oral glucose tolerance test. And it's that bookmarking and also the quantitative assessment of stage 1 or stage 2 that has been really helpful in the staging paradigm. And stage 3 is actual clinical onset of symptomatic type 1 diabetes. With each stage, they have this research base has set the stage for development of therapeutics intended to delay progression. For stage 1, we know that they have a lifetime risk of developing type 1 diabetes. So 100% will go on to develop type 1 diabetes. But once they develop stage 2, it's extremely variable in their rate of progression. But once you develop stage 2, you have a 60% risk of developing type 1 diabetes and needing insulin within 2 years, and then an 85% risk of needing insulin within 5 years. And so when you're looking at a drug that has risks and trying to form your benefit risk assessment, it's really helpful to know how the length of time that the patient's going to between the treatment and developing type 1 diabetes. And it helps you weigh the risks of the drug against the risks of actually developing the disease. It's much more acceptable to have a risk if you're going to need insulin within 2 years. And so one of the initiatives that CPAC has involved our division in is a way that they interface with the agency multidisciplinary groups called a Fit for Purpose initiative, where they develop a biomarker of sorts, but a clinical trial simulation tool that Klaus was kind of harkening on. And my experience with this initiative has been, it's in its process right now, but they're developing a clinical trial simulation tool whereby they're looking at predictive biomarkers for type 1 diabetes progression to help design efficient, effective clinical trials. And one of the important things that has come up over and over again is disease states that extend the lifespan, pediatric to adult, often present more severe, more rapidly progressive in children. This makes sense. Children, if you have a genetic defect that leads to disease and it comes in childhood, you likely have a much more rapidly progressive disease course than if it presents in adulthood. And thus, enriching your trials with people who might be more rapidly progressive, knowing different genetic markers that can really help you predict, helps us from the agency standpoint with the reliability and our benefit risk assessment, which at the end of the day is what we're we're had the decision we're trying to make. And so the Fit for Purpose initiative kind of brings it in at an early stage, allows all of our experts to comment and give feedback, and also help us to get our regulatory questions answered at an early stage.
That was fantastic, Lauren. Thanks for the very comprehensive and clear linkage between a very robust previous research to understand staging, and then do more with the staging approach and understand those time varying probabilities of crossing each one of the stages, but within the stages, looking quantitatively as how the dynamics of within the stage, the metrics behave. I think that's a really important point to highlight, because in a high collection of neurological indications, very similar attributes to the type 1 diabetes staging approach are now something tangible and something real. Started with the 2018 guidance for early drug development for Alzheimer's disease. Now the Huntington's community has developed one. Parkinson's is getting started. The spinocerebellar ataxias are getting started. And so that's a key opportunity that could be explored with many diseases, pending, of course, the availability of data. But I want to get back to something that you mentioned around the types of discussions that happen with the agency, with all the relevant groups to understand what are sometimes very complex, nonlinear relationships between different metrics. And I think that's the point that Celia, you were raising about, okay, we really, the one-to-one relationship in a straight line might always be the case. And so, Jeff, I want to come back to you because you saw some of that happen in a rare orphan kidney disease and how the journey happened with the agency there. So I wonder if you could also offer your perspective of how that journey went and the discussions that happened as part of that effort.
Sure. So I think what Klaus is referring to is work done with autosomal dominant polycystic kidney disease and the way the biomarker was developed of CT-based kidney volume. But let me step back. So you heard an excellent example from Dr. Heiickman of finding a prognostic biomarker that allows you to select patients who in a relatively short period of time will develop a clinically meaningful endpoint, development of diabetes. So you can design trials of, for example, 2 years in duration to be able to see how effective a drug is at preventing development of diabetes. With other clinical outcomes in other diseases, the meaningful clinical outcome may be many years out. Examples of this are non-alcoholic steatohepatitis, for example, where cirrhosis and liver decompensation provide adverse clinical outcomes that may be 5, 10, even 15 years out. So you really can't conduct a controlled trial 15 years in duration. So in situations like that, the FDA considers surrogate endpoints as reasonably likely biomarkers as reasonably likely surrogates, or in some cases, validated surrogates for approval of drugs. So for NASH, for example, non-alcoholic steatohepatitis, liver biopsy is used as a reasonably likely surrogate with a clinical trial to be, and that's used for accelerated approval with a clinical trial continuing on to look at those clinical outcomes that are further out. In developing those surrogate endpoints, it may not be possible to do the study just like that because the level of evidence sometimes has to be very high. So in the example that Klaus cited, autosomal dominant polycystic kidney disease, the biomarker of CT-based kidney volume was assessed as a prognostic biomarker in a consortium led by CPATH. This biomarker was shown to be prognostic of development of chronic renal failure, and a model was put together identifying the key characteristics that would show which patients were likely to progress in 3, 4, 5 years. This was qualified as a prognostic biomarker by the FDA, and then it was incorporated into clinical development programs and demonstrated to be associated with long-term clinical outcomes, actual development of renal failure. Based on that, the total kidney volume is now accepted as a reasonably likely surrogate for approval, for accelerated approval of drugs for autosomal dominant polycystic kidney disease. So this is an example of a stepwise approach to developing a very impactful surrogate endpoint biomarker. Celia and I are working with CPATH on the alpha-1 antitrypsin deficiency consortium, where we hope that similar data can be pulled together from a variety of sources to define whether there are biomarkers that can be helpful in that disease as well.
Yeah, that's fantastic. And I think the way you linked the points is very, very illustrative to the kind of vision that we're having for how to structure things and the journey that we're planning to take in alpha-1 antitrypsin deficiency. So I'll probably go to Celia to have a last word on anything that you think that we may have missed in this illustration of how important it is to define the specific use of a biomarker and how specific uses may have more impact than apparently at first glance, and how to drive things progressively forward?
That's also a pretty broad question.
Yeah.
But I just want to just take a step back and say, the first thing is to describe the biomarker in enough detail that, we understand its characteristics as a measurement tool. And that's almost irrespective. But I think that's one of the challenges in aggregating data, as CPATH is so good at doing. But I'm hoping that, there'll be a way forward looking at this, and we'll be able to, make some sense out of it. And I think one benefit of the kind of thing that CPATH does is not just to focus on one data element, but to look at, all the information in some of these collections of data, which are quite rich, some of the sources of data, to try to see whether there's some insights that emerge.
Fantastic. Joe?
Yeah, I want to thank again our panelists and the audience here today. I think one of the things that you have heard repeatedly is this idea of engaging early around these discussions around biomarkers, having this clear concept of the context of use, being able to have those conversations. The earlier you start, the better off you're going to be. It gives you time to adjust. You don't want to walk in thinking you've got everything figured out, only to have 3 or 4 new questions posed to you, and you're now looking at another decade of work. So I think I want to encourage everybody, regardless of whether you're working with Critical Path, which I would obviously recommend, or with others, please start that process as early as possible and engage the regulators as early as possible. So thank you very much.
Thanks, everybody. Early and often. I think we were able to get us even ahead of time, so we're going to take a break now, and then we're going to reconvene for the last panel of the day. Thank you so much. Thank you. So,
Very nice.
no, that was nice. Thank you.
Really,
Yeah.
really.
He is.
It made the point, like,
Oh, yeah. No, we
You're in person finally.
Yeah,
First time in
I thought that
person.
was a good count.
This was great. I love it. You guys were so efficient, This is awesome.
Did I? Okay, great.
Yeah, it's very good.
Thank you for introducing yourself.
So yeah, you've met pretty much all the team in person now.
Hey, I'm really excited. Phase 2.
Yes. Yes, and but as you say, now we're going to really get to that's what I'm really excited about.
Did you get to listen in on the consortium meeting yesterday? No, it's okay. I just was wondering if you, because it was really an amazing experience. Yeah. Okay. Yeah. Okay. No, it was. I mean, you live this all the time, but we don't. We're in a room as FDA, it's a room full of pharmaceutical company executives. And
Yes.
they are eager to hold resources to develop new drugs for conditions that they just want to know what they want to do. So, yes.
You're gonna get to experience this because that's the life.
It came up over and over. There was a woman, I can't remember what that company she was.
CSO Mary.
So she said,
What?
Oh, right.